in Erasmus MC, working on difficult to treat infections. In particular, we are very interested in how to treat chronic infections. Often they are associated with biofilms, and biofilms, of course, are well known to be causing big problems in so the immune system cannot handle them. Sensitivity towards antibiotics is also pretty low. Furthermore, biofilms are well known to cause secondary infections. And we put a lot of medical devices in people. So there is, if there is a biofilm on a life-saving implant, they need to be taken out, which causes, of course, a lot of trouble. Mortality and morbidity, but also hospital length stay and increased healthcare costs. Examples in the hospital of bacteria that cause issues are Staphylococcus aureus, Staphylococcus epidermidis, Acinetobacter, and Pseudomonas aeruginosa. There are, of course, several others, but these are important ones. When we talk about biofilms, I'm talking about planktonic bacteria that adhere to a surface. This can be either a polymer that we put in a person or natural surfaces. Bacteria can adhere and form clusters, encapsulate themselves in bacterial proteins, human proteins, bacterial and human DNA, but also by the bacteria produced sugars. Finally, ending up with mature biofilms like this. And at a certain moment, they can disperse and their secondary infections can appear. When we look at Staphylococcus aureus, there's another issue here, and that is that bacteria in biofilms get exposed to antimicrobial peptides, and there is a component system that is able to register these antimicrobial peptides, and as a consequence, alanines will be placed on the liponucleic acid and lysyl groups on the phospholipids layer, making the charge of the bacteria slightly more positive, causing repulsion for the antimicrobial peptides. Unfortunately, antibiotics often use similar kind of ways to get into the bug. So a positively charged, an increase of positive charge either absorbs the antibiotics or repels them, making biofilms intrinsic less sensitive, up to 1,000 times less sensitive for antibiotics. In addition to these changes in charge on the direct surface of the bacteria, Staphylococcus aureus is also well known for generation of a layer of PIA, which is basically an N-acetylglucosamine. Deacetylation also causes the sugars to become more charged, and over here you can see what that has a big impact on the positive charge, repulsing, of course, antimicrobial peptide, but also antibiotics. So how can we, in a reliable way, quantify the impact of antibiotics on biofilms? And I don't think I have to tell this audience that classic way of looking at biofilms causes issues. Crystal violet assays are often quite unreliable because of reproducibility. So, and here we are instantly at a big problem, and especially when you work with bacteria that cause agglutination. And I will show you a couple of examples. If you have a bacterium that causes that has loose colony of loose bacteria in a planktonic device, this will have a certain OD. And but if the same amount of bacteria cluster, then of course the OD will be less optical density. When we look at the CMU, This is of course 5, but over here you will get 1 CFU. So you easily underestimate, in this case, the amount of bugs. If 5 bugs, for instance, divide, but during the division of 1 hour twice, they remain sitting on alongside each other, like in case of staphylococci, your optical density during this hour will slightly increase. But for instance, your CFU won't. So again, there is an issue. How do you measure the amount of bugs? Another example, if we have 5 bugs that remain clustered after the division, or the same 5 that spread out. I hope you appreciate that when we start with a CFU of 5, the optical density in this case will slightly increase, but the CFU won't. And over here, you will get an increase in your CFU of optical density, but also your CFUs. So, but in our case, we often don't know, and especially if you study Staphylococcus aureus, are we dealing with bugs that look like this, or are we dealing with bugs that look like that? On top of that, if I have, for instance, my 5 bugs, my 5 CFU, treat them, and in one case, for instance, allow the bugs to lyse, then I will have a reduction in optical density and also a reduction in CFU. Yet, if the bugs agglutinate as a consequence of the treatment, then my CFUs also will reduce. My optical density will reduce in more or less a similar way, but my CFUs also. So if in this case, I might say, hey, the bugs, I have a very active component that does something with my bugs. So if you work with Staphylococcus aureus, this is actually constantly happening. And then if you think of how to work with biofilms, this increases. So a couple of years ago, I was exposed to isothermal microcalorimetry. And in this case, of course, bacteria generate metabolic heat. And this machine is so sensitive that roughly 10 to the 4th bacteria and the amount of energy can be detected. So could it be that if we treat a bug and they lyse, that the energy is reduced, and if they agglutinate, that you will see this? I can confirm yes. So using these strategies, you can determine in a much better way how much bugs are there. And now I would like to return to our biofilms, because in case of biofilms, this is quite a nightmare. I hope you can appreciate this. What I now depicted in planktonic is, of course, happening all the time in a biofilm. So after quite some fiddling around, we came up with the following assay. This is the heat flow, so this is the amount of energy that is generated by the bacteria. And when we grow Staphylococcus aureus, not in classic bacterial growth media, but in IMDM in this case, our reasoning was human cells. We would like to make a human mimic. Human cells won't grow in typical soya broth or in IMDM or in other bacterial media, but they grow in IMDM. So let's see what the bug does if we expose them to IMDM. And initially, you see an increase in growth, and then after roughly 10 hours, there's a peak, and then it shallows down. When we took the vial out, and hope you can appreciate, there are indeed clusters in the bottom of the small vial. Then we took the vial, exposed it to antibiotics, and in this case, rifampicin, and then you can clearly see that the amount of energy reduces dramatically. We took this data and normalized it for our control, and then, of of course, you get these kind of curves. And from now on, I will show you data generated in this normalization. So we took a whole panel of MSSAs and MRSAs, and here you can see the different genetic backgrounds, because we used representative genetic background of Staphylococcus aureus in our MSSAs, but also in our MRSAs. And these are the Vitek data. So these MSSAs are very sensitive towards rifampicin. These are the mix. In case of these MRSAs, you can see that mu50 is quite resistant to rifampicin. Here you can see our plots. We took, in case of, as a control to compare these two figures, the resistant strain, the mu50 strain, they both are plotted. And I hope you can appreciate very, very quickly, you can see that the energy is reduced dramatically when you expose the bugs to a concentration that is maximally reached in human serum. Unfortunately, in case of the MSSAs, already within 40 hours, you can see that there is an increase of bacteria again. We took these bugs and analyzed them, and indeed, these are resistant. But the ones that didn't become resistant, and these are not resistant, became more or less tolerant. And the MRSAs and the MSSAs more or less behaved in a similar way. So most bacteria are affected by rifampicin, yet they're not dead. And you can culture, we cultured them after the incubation in this machine, and then we could clearly see vital bacteria. Unfortunately, in 2 cases, we see resistance popping up already within 24 hours after treatment. In case of, for instance, endocarditis, flucloxacillin is often used. So we wondered what happens when we treat our panel with flucloxacillin. And this is, again, the concentration that is used in case of the maximal serum concentration. Here you can see our Vitek data. These strains are very sensitive, MSSAs, and in case of the MRSAs, all strains were resistant, which you would expect. This is the mu50 again, and I hope you appreciate that indeed the MRSAs are not affected at all, or slightly. This is our. But when we look now to the MSSAs, it's quite shocking. They are not, and some even outcompete our resistant MRSA that are depicted over here as well. So, unfortunately, regardless of being an MSSA or an MRSA, according to the classic approach, biofilm-associated Staph aureus can survive concentrations of flucloxacillin that are maximally reached in blood. Why is this? Vancomycin is, of course, also often used in clinics to fight, in particular, MRSAs. And when I now look at first at my MRSA panel, they're all sensitive, except for mu50, which is considered to be an intermediate. Look at our profiles. The intermediate strain is actually doing the poorest when we look to the other MRSAs. When we look to the MSSAs, more or less, except for strain CC30, are also similar and can tolerate vancomycin in a similar way in comparison to a strain that is in the Vitek considered to be intermediate. So in our experiments, biofilm-associated Staph aureus have high vancomycin tolerance. We also, in this paper, studied other antibiotics, and here I depict 2 strains, one MSSA and one MRSA. And when we again look at the MRSAs, I think it's very clear that Vitek data resistant. These are the biofilm sensitivity for these, so they are indeed resistant. Yet, if the Vitex tells they're sensitive, we see an effect in the biofilm. Unfortunately, they can start tolerating after some time higher concentrations, and we see even the energy going up, indicating that the bugs start dividing, maybe. That is something I'm not sure currently, but they're not dying. Over here are the similar antibiotics, and of course in MSSAs. For this MSSA, the Vitex data are showing that they're all sensitive. Only in case of the rifampicin, we see a reasonable reduction, but resistance within 24 hours. To indicate that it's not only, how do you say this? I don't want to end up this story with just bad news. We started combining the antibiotics, and here 2 examples of 2 strains. We combined flucloxacillin with rifampicin, but also independent. So this is the rifampicin. After within 24 hours, we see in resistance, and in case of flucloxacillin, there's more. But when we combine them, then it's very clear that the resistance in the period that we were measuring didn't appear. Combination of levofloxacin and rifampicin, we can see the same. Though the independent antibiotics, either they hardly don't do anything, or resistance appeared. One combination of vancomycin and rifampicin is the same. This is one strain, and this is an example of another strain. And here again, clearly, it looks like the combinations that I just mentioned. So with the fluclox, with levo, and with vancomycin, at least I don't dare to say prevent, but during the time that we have been measuring, we don't see resistance strains, rifampicin resistant strains popping up. We also found combinations that are less well. So here is one strain, and here's the other strain. When you combine clindamycin with rifampicin, this is the line for clindamycin alone. This is the profile for the rifampicin alone. Look, when you combine it, rifampicin loses its activity, and that's also count for when we do this for CC27. 72. The rifampicin effect is not there anymore, and you can only see more or less the clindamycin profile. So it looks like this is either you can antagonistic, or something like Clinda takes over. I'm not really sure. Coming to my first conclusions, biofilms of MSSAs become an MRSA, at least under the conditions tested, and the strains, of course. Biofilms that we tested can tolerate high concentrations of vancomycin. Combination of rifampicin, flucloxacillin, and vancomycin with prevents rifampicin resistance. Rifampicin in combination with clindamycin shows antagonistic effects. Beside the paper that we sometime was it in 2022 published, we are currently working also on LVAD driveline infections. If someone's heart is failing in the left ventricle, a pump can be placed, and this pump takes over the function of the heart because it will pump the blood into the aorta via this pipe. Unfortunately, this pump, of course, needs power. So until here, this is sitting inside the human, and here it sticks out, and this is sitting a machine that is sitting on your belt. These batteries are, of course, the battery stopped, the person will die. So this is how a driveline looks like when it sticks out and it's not infected. Unfortunately, this is an infected driveline. 60% of the people that have an LVAD heart pump can survive up to 55 years. So it's quite a nice way to if a person needs to wait for a heart transplantation. Unfortunately, if your driveline becomes infected, the chance that you will get a new heart reduces. So we wondered, can we mimic in our system these driveline infections? So we took drivelines, cut them in pieces, and grew for 3 days biofilms. And then at the last day, the 4th day, we placed them in the calScreener to see if we had enough energy and exposed them to drugs, and then allowed them, got rid of the drugs to see if there's regrowth. This is basically the control. So this is the after 3 days, the 4th day, fresh media, and you can clearly see that there's a nice profile. And this is not a triplicate, but a quadruplicate. When you expose them, of course, to no antibiotics, they go up again, and of course, regrowth is there as well. When you increase the concentration of rifampicin during this time, the profile changes dramatically, and already at 0.3 micrograms, it's just depending. And the curves start to fluctuate quite a lot. When we increase the concentration of rifampicin to 0.5 micrograms per ml, it is very clear that the bacteria are killed because there is no regrowth. So we think we can monitor, using this, we can monitor antibiotic sensitivity of biofilm-associated bacteria on drivelines. And we hope that this application in the future will help us to design personalized strategies to combat these driveline infections. We already did this. It works also with Pseudomonas and Acinetobacter. So, and these are often seen, at least in our department, in patients with these driveline infections. Just for the last couple of minutes, I would like to tell you something about. Of course, this is the part that I showed where biofilms are formed on a surface. Yet there are, of course, also a whole lot of bacteria that form aggregates, and Azorius is one of them. There is a debate: can we talk about biofilms, yes or no, when they are planktonic or even on the surface, because that's also happening. Pseudomonas can do this, but also different microbacteria grow very nice floating devices even, and you can really see them in formal biofilms. So we are in the lab working on how can we catch these biofilms and do assays with them. So one of my technicians, Corné de Vogel, and Michiel Bekskens, designed in our 3D printer several, and here are examples. In order to catch the biofilms, and currently we can really generate these biofilms in a 16 well plate, a 96 well plate, and transfer them to calScreener. Either biofilms that are formed on the surface, or biofilms that are floating or are formed on the air-liquid interface. So I'm convinced that we are able to monitor the behavior of biofilm-associated bacteria in a way we were never able to do. And therefore, we obtained a set of new eyes that allow us to observe the unknown. I need to mention a couple of people that are very much involved. Nicole, Mary, Corné, Rofjan, Michiel, and yeah, that is more or less what I wanted to say.
Thank you very much, Willem. I don't suppose you can hear me now. Can you hear me? Okay, so for anyone who has just joined us, then we have had trouble with the audio. So it's going to be very difficult to do a Q&A actually, but we are going to try. So if you have any questions here, please try type them in the chat. We've also got...
I don't hear anything.
Okay.
I'm not sure why I don't hear anything.
Can you... Let's have a look at this now. So I'm just going to type to Willem in the chat. Can you see my...
Would you recommend sonication for better quantification? Unfortunately, sonication is killing bacteria on one hand, and how do you control in a proper way on still aggregates? It's very, very, very difficult, and especially when you look at biofilms, and when we look at biofilms that are in vitro generated, they often easily fall apart. But when we look at biofilms that are generated in vivo, they are encapsulated in bacterial components, but also in case of, for instance, in blood vessels, in fibrin. So and bacteria can bind to these fibrin particles like no others. I mean, as always can do this. Therefore, we have a big problem I'm pretty sure that you might underestimate the amount of bacteria. And especially if you start doing CFU analysis after sonication. So, question how really... one question I have related to the potential detection of persistence. Yeah, of course, persistence are a big issue. The problem is, no, I should call this in a different way. When we started this work, the first thing was how much of the persistence are there in biofilms. Actually, in our hands currently, and especially when we talk about Azorius, we don't see so much persistence. We often, the amount of energy that is generated by these biofilm-grown bacteria is usually lower in comparison to when you do similar kind of planktonic experiments. So it looks like the bugs scale down their metabolic activity in the biofilm, making them, of course, less sensitive to antibiotics. What we always do in order to prevent missing out on persistent cells, after treatment with antibiotics, we grow them on blood agar. So there you can see how much bugs are still revive after your exposure. Are there interesting. Are there any recommendations for dry surface biofilms? What we do, we have been doing quite some work, and that's the work of Valerie Bade. Using strategies so a recent paper published by our group, we used coupons in which we allowed the bacteria to dry out, and then put them back into the calScreener without having to determine CFU nor optical density. Could determine by the lag time the amount of surviving bacteria. So I think yes, no, sorry, I know that you can easily determine the amount of bacteria sitting on a surface using strategies that we describe in that paper. Yeah, so that's interesting. Multicellular species. A couple of months ago, we had a drive-line infection of a patient, straight coming from a patient, and we knew that there was a Pseudomonas. So we took this piece of a drive-line from the LVAT and we placed it in our calScreener. And what we saw, Pseudomonas very quickly grows very significantly. And in another tube, at a certain moment, we saw another peak coming up. When we looked at our plates the next day, we could see that there was a Pseudomonas on one hand and a Serratia on the other hand. And looking at these peaks, most of these bacteria, and especially when they're allowed to grow planktonic, show a species-specific profile. So if you allow them to grow again on in planktonic conditions, you can get an idea on what's going on. Following up, yes, sorry, persistent. So some of your work shows that you removed the antibiotics, then allowing bacteria. Yeah, this is exactly the persister issue, and we always do that. There is no other way, of course. One of the discussions is, of course, also how much bacteria do you need to kill, or do you need to, how much reduction do you need to have in order for the immune system to do something? And yeah, that's only the future can tell. I don't know. And potentially the persistent cells start growing and generate new more active cells. Well, if you have these more active cells, then your antibiotic treatment can be done again. Yeah, so a long-term approach probably, and that's of course also what we see, need to be approached. Those are... Oh yeah, thank you. So how much, in order to prevent a big impact of planktonic, we of course wash them. Yeah? So the very first couple of hours, the amount of planktonic bacteria is very low. Another thing is that in most cases of most of experiments, planktonic bacteria cannot handle the amount of antibiotics that we apply, except of course for the MRSA in certain conditions. So we are pretty sure that most of our signal comes from indeed biofilm associated bacteria. Yet there will be We cannot say that there are no planktonic bacteria. I fully agree. Also, I think that yeah. How long was the duration? This of course depends. Sorry, how long was the how long did we the intermediate period? Well, after the overnight incubation in the calScreener to determine are all the biofilms growing in the same way, you take out Cornet took out the supinate to get rid of all the planktonic bacteria, and then replaced put in new media with antibiotic, yes or no. This is done as fast as possible, but well, it takes something like half an hour to do this. And then, of course, you have to recalibrate. So then it takes roughly 40 minutes in order to start being able to measure. That's why a part of the rifampicin data, we miss the first drop. Yet, when you compare it, of course, to the untreated, you can still determine what is the effect of your antibiotic. That is, of course, a disadvantage of the system. On the other hand, if you design your experiments for the long term, and in case of biofilms, you can do this because, well, they sit there and stay. This is, in our hands, not the most difficult issue. One of the difficult issues is, of course, we cannot change the amount of oxygen in there. In case of oxygen-dependent bacteria, this can be an issue. Pseudomonas is one example of that. Also, some microbacteria are suffering under this. But when you work with Staphylococcus aureus, you actually prefer that the oxygen is decreased. Why? The conditions in humans, there where the bug forms biofilms, are often not oxygen exposed. So it depends on the bug, and it depends on the conditions you want to work with. Okay.
Thank you, Willem. We persevered there with lack of sound.
It was nice to be here, and I'm happy that we were able to. I was able to show our data. Looking forward.
Thank you very much, and thank you for everyone who attended. You'll get email link to the webinar replay. So thank you very much indeed. Enjoy the rest of your day.